record on this computer.
Okay, you're on.
Perfect. Thank you. So, as I said before, in addition to the type of capturing modality that you're using or the type of machine that you're using, another parameter that is very important to for clinical application was the type of sequencing modality, which at the very end was a single read. So sequences were read only from one direction or the other direction. So we had only a single read analysis, and then the content was produced. But it has been seen very early on that there are events where, for instance, translocation and there are fusion protein, fusion gene events, where you could have one sequencing from one side which matches one particular gene, and the sequence from the other side of the product, the fragment that you were sequencing, was coming from another part of the product, and so people got confused. So the pair-end technology came to rescue, and right now the most common read length that is used clinically is 150 base pair end technology. There is another way to detect variants or events in the large portion of the genome, and this is the mate pair, and it works. It was used to work in a way that you fragment your DNA of interest, then you circulate, you circularize after biotinylation, pull down after enrichment, and then those fragments are fragmented again, and they are treated as the regular fragment for sequencing. And those approach have been found to be extremely useful in the fusion gene disease, like for CML or chronic myeloid leukemia, where and ALL, that is acute lymphoblastic leukemia. ALL is a frequent cause of leukemia in kids. Usually they have a favorable prognosis. If ALL occurs in adults, the prognosis is dismal. So this fusion occurs between the two genes, BCR and ABL, and of course causes a new product that has new function, so it's a gain of function. And this large product detection can aid to identify those particular fusions. And in this case, it's very important because this fusion product is can respond to therapy with a humanized monoclonal antibody called imatinib or Gleevec. So another event is in acute promyelocytic leukemia, or APL, where we have the PML and the RARA fusion. And also in this case, it's very important to detect what type of event has occurred and in specific details, because this can tell you whether the patient can be treated with retinoic acid because it is responsive to retinoic acid or is non-responsive. So the prognosis is going to be different. Okay, so we have chosen, the type of target we want to do, the type of capturing modality, how long the reads have to be, the type of machine, and now we have the sequence. So this is your realm now. So that's the sequence that we usually, yeah.
So for PML RARA and CML transplantation, wouldn't it be easier to just do a real-time PCR assay instead of doing a sequencing?
Oh, yeah, yeah. It can be done by real-time PCR. It can be done in many ways. It's not that I'm just saying that this is another application of the massive parallel sequencing, which can be done. So with the made pair. So the made pair is detecting gross. Right. 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 Exactly.
Implement those for treatment because in PML you're looking for treatment like a day of like once you do the analysis.
Yeah, depends on the technology you want to apply. Usually, real-time PCR has been, at least in the recent past, much faster than next-generation sequencing. But depending on the level of automation of the laboratory, next-generation sequencing can be done on a stat level, so short turnaround time. Just to give you an example, there are even uh, academic institutions like Kansas City or other academic institutions that can provide whole genome sequencing with a 3 days turnaround time. So true that technology may change the way that you choose what type of test you want to do, but is in reality is not only the machine, but it's everything that is implemented. So the level of automation in terms of sample processing, sample handling, sequencing, and then the throughput analysis. So there could be a lot of implementation in bioinformatic algorithms to go much faster. And if you know exactly what you're looking for, then even the sequencing technology can be as fast as a real-time PCR, and definitely sometimes more accurate. So those are the type of let's say data that you receive. So the first line just indicates the sequencer, so the machine that you have used. The second line is the raw data, the raw sequence with the letters, and those are all matrices that you will have to record for clinical purposes, because you need to know if something went wrong. So what was the machine where it went wrong, and even later on, what is the position of your plate. And so that's why you have not only the unique instruments name, but also the flow cell lane. There are instruments with only one flow cell, so it doesn't matter, but are other instruments with multiple flow cells. And so that's also very important. The tile number within the flow cell lane, so exactly you can pinpoint what went wrong there. So all those metrics are extremely important and needs to be recorded. I mean, they are in the file, of course, but this is a spit out from the machine, but still you need to retain those data. One thing that everybody needs to keep in mind is that when we have a sequence, every letter of the sequence will be paired with a certain symbol. And this symbol represents actually the quality, so how good quality for each data point has been obtained. So the exclamation mark represents the lowest quality, while this symbol represents the highest quality. And then everything is in between. I don't know exactly how all those symbols have been, selected, and
they know.
Okay, perfect.
We'll be testing them whether they know or not in 2 weeks.
I will be very curious. Yeah. So, and then we go to the bioinformatic analysis. I will go very fast because otherwise, you will outperform me in this. But definitely, the most important thing to keep in mind is that the machine will produce a so-called FASTQ file, and then obviously you have to go through all those different softwares, and everything is changing. So this is an old slide, so probably it's already outdated completely. But and so the most important thing that that you need to keep in mind is that the most important thing to obtain is the data where all the sequences obtained from a clone will be then put together eventually in a contig and will be mapped against the reference sequence. So you need to know not only what every fragment that you have been sequenced contains in terms of sequencing, but also where it maps compared to the reference sequence and all the changes that occur in those fragments that you have sequenced. So one part that is very important, and this is an old slide with the coverage analysis, is to know how many times you have covered a specific nucleotide and or a specific target for that matter. So this is a coverage analysis where where roughly 95% of all the target have been covered 50 times or more. So this is a very good coverage, but this was a 300x average coverage run. And that's what you should expect in a clinical grade sequencing, at least that.
Even for whole genome?
No, for whole genome, the clinical coverage, even though the limit was set at 40 as a minimum.
Okay.
Average coverage. 40x unfortunately is suboptimal. You should look at the 80, 75 or 100x, and the minimum 150 for exome. Well, if you want to detect, in addition to sequencing, also other events that I will show you later. So not only the coverage is important, so how many data points you have per position, but also you want to look in this IGV or integrated genome viewer analysis if you have coverage from both directions. So this arrow shows that you got reading from this side, this arrow from this side. So depth of coverage is important. The change where it occurs compared to the reference sequence. Here you have the chromosomal position, where the gene sits and so forth. And here you have all the variants that have been detected. It is very, very important to always make sure that you have reading from both sides, from both directions. It's a bad sign when all the readings are coming only from one direction. Usually they're not acceptable clinically.
It's particularly bad if all the variants are from one strand.
Correct. But even all the reads are coming from one strand. You want to see reads coming from both strands anyway.
No, for your PCR-based assay, sometimes it's not possible, right? So if your capture part is using PCR, and then for this end, you can only have one direction.
If you're using an amplicon-based,
Yeah,
sometimes you don't have a lot of choices.
right.
But if you are using a hybridization base,
You want to see both.
You want to see both. So of course, you want to call the variant, so detect the variant correctly with a good quality, so that statistically it will stand. You know, that this call has been done in a correct way, where you have a lot of data per data point. So and you compare that to the reference sequence. So you need to have a high coverage, you need to have high quality per coverage. Sometimes if you have a high quality detection, even a lower coverage than the average may be acceptable. But if you have a high coverage but with very bad quality, then this is not acceptable. So it's better to have high quality and a little bit lower depth of coverage, but still higher than the minimum that is considered in clinical application. Between 10 and 20x is the minimum you should have in order to make a call. Minimum.
So you prefer to have a more coverage, even though the base quality is a little bit lower?
No, I prefer to have a higher quality per base, even if the coverage is a little bit lower. let's say
I see.
a 50x coverage with a high quality will give much higher confidence than a 2000 coverage for extremely bad quality data point.
I mean, the reason I'm asking this is a practical issue is if we have the set budget and we can do either 2x75 protocol or
Yeah.
2x150 protocol. If we do the second one, it will be cheaper, so we can afford to sequence more. The coverage will be higher, but towards the end, the quality will drop.
Yeah, if you do the shorter read, it gives you higher coverage, but then you're going to be with less quality, especially
If we do shorter reads, and then our average coverage will be lower with the same amount of budget.
Oh, well, but usually that's they did 2x50 rather than 2x150 because the more reads usually leads to less coverage per point, meaning that that's what they were doing. Meaning at the beginning they were doing short reads because it was giving a lot of coverage because the output is fixed per machine. So if you have for instance, a 15 gigabytes output, gigabases output, then with shorter reads you have more coverage. With longer reads you have to sacrifice the coverage, but you have a much better quality in terms of detection of longer reads and differentiating between real target and pseudo genes target or other targets. So that's why you usually go with the longer reads in order to be more specific when you map to the reference genome. That's usually has been the case.
But towards the 300 cycle, and the base quality will go down.
Yes, yes. So you're looking at the first, let's say, 75 to 100 cycles are going to be your gold mine, and then at the very end your quality may go down. And yeah, because the reads start to be a bit,
the reagent already
there. Yeah. So then of course, in addition to the variant calling, so having high quality, high coverage, and determining the zygosity, remember, not always, even for constitutional genetics, you will be always, lucky if you always find 50-50 ratio between the variant, but everything that is close to, can be called confidently. When you have also to determine, when you are in a clinical laboratory, what is the minimum to call heterozygous versus calling homozygous. And the difference between single nucleotide variants and smaller, larger indels, it's great because when you have lower quality, then also your ratio to call heterozygous or homozygous has to be adjusted accordingly. The other thing is annotating the variant, so identify the specific type of variant, if it is a missense, a nonsense, a frame shift, whatever, silent change, and the biological significance. If it's changing an amino acid, or if it is at the very end of, for instance, an exon, even if it's silent, so it may have still a biological impact and so forth. There are also a lot of in silico algorithms for prediction for missense variant, especially for missense variants, so if they can have an impact on the protein. There is a limitation, is that if you have a protein that binds another protein, and this is the vast majority of what happens in biological function, there are very few proteins that are just floating around by themselves. Those prediction software, they don't predict the effect of that variant on the binding partner. They just predict what happens to the protein itself. So, and another thing is, it's extremely, even more important to have up-to-date the reference sequence and all the possible isoforms. Because, when you do a massive parallel sequencing, you can detect all the possible isoform, all the possible exons, coding exons in the gene. Let's go to Titin. Titin has 300 and 64 exons, 363 are coding. Of those, 363 are making what we call a meta-isoform or meta-transcript. This is not existing in, so does not produce a biologically functional element protein. So, but there are other isoforms, a lot of isoforms that are alternatively spliced, which produce a biologically functional protein. And so even though you want to capture all the meta-transcript, really biologically you don't need to, because there are a lot of exons that are not expressed in your organ of interest. Post-bioinformatic analysis, that's what I used to receive in terms of data. Yulong knows extremely well. I don't know why it doesn't really show very well, but anyway.
Because we generate. So that's why it doesn't
No. Meaning on the screen, it seems to be low quality image, and I apologize for that. But essentially, you receive in this case, is an Excel file with a large amount of data that you need to analyze. Here it's a little bit better. So you need to know, of course, the chromosomal position, and so the relative position, the position relative to the genome where a variant occurred, and then it would be great to link to several databases. So to link, for instance, to a database called HGMD, or Human Genome Mutation Database, which tells you if this particular variant has been published before. And so if there are literature attached to the variant, it's important because you can go to that piece of literature and analyze the evidence that are presented and see if they are helping your classification. The other thing is, obviously is key to have the gene of interest and the type of variant that occurs. In this case, is a missense variant. And I think, even though you cannot see and I cannot see, but I believe is a glutamine 403 to arginine change, rather arginine 403 to glutamine change, which is the most common variant in myosin heavy chain 7 causing hypertrophic cardiomyopathy. So there are a lot of databases that you need to pay attention to, and a lot of parameters. So those are the parameters of the quality, the zygosity here. So you want, for instance, for an autosomal dominant disease to see a heterozygous occurrence. And then the depth of coverage is extremely important. So this tells you how many reads you have per this particular position. If it is in an exon or 5' UTR, what type of change it is, the gene. And in a clinical setting, whenever a variant has been identified and needs to be reported, it's always good to, actually is required to confirm this variant with an orthogonal technology. So the orthogonal technology, once you do next generation sequencing, the first orthogonal technology that was used was Sanger sequencing. So getting rid of Sanger is going to be hard. But other technologies has been utilized, like other massive parallel sequencing ways have been utilized. Or sometimes if there are recurrent changes that you detect, you can have a Tuckman probe or SNP array or whatever you want to use, depending on the application. It is also important when we detect the variant in order to provide the classification to determine if this variant is present in the general population. So there have been many databases historically. So the DBSNP, the 1000 Genome Project, the ESP database, then the EXACT database, and now the NOMEDY database, which includes not only exome sequencing, but also 15,000 genome sequences from general population. In addition to that, so once we know if the variant has been identified or not, before prior, has been identified or not in the general population, then we need also to determine if it has been published. You can even Google it, but PubMed has been an invaluable tool for that. And also you can use all the prediction algorithm in order to determine a possible effect on the protein. So once these data, so we have identified the variant that it's potentially important, then we need to get to determining and weighing all the evidence that we have in front of us. So the clinical interpretation of a variant is determined by the type of variant. Is it fitting the mechanism of disease that has been established for that particular disorder? Yes or no? Then if we have a family studies, so if the patient has a family history of the disease and there are multiple affected members of the family, then it's advisable to really test all those family members in order to determine if the variant segregates with the disease. And so, because having, especially with multiple generations, you generate a statistical power where the variant cannot occur just by chance, but has to occur because it's attached to the, it's associated with the disease. Extremely important, the population genetics, in addition to those databases, also are very important, those GWAS studies that has been done across the globe, where large cohort versus, case versus control analysis has been done for many disorders. And in some cases, there are variants that can be more present in cases than controls. And this has to be taken in consideration for interpretation. Unfortunately, functional studies are almost relegated to research laboratories, mostly to research laboratories, and they're beyond the scope of clinical labs at this particular moment. So variant types, when we find that there is a certain type of variant that occurs in a patient, we may get excited sometimes too soon. So we have always to keep in mind, the mechanism of the disease. So when we, for instance, identify truncating variants, those are nonsense frame shift of splicing. When, for instance, we have splicing variants, first of all, we need to determine whether they are in the invariant position, where the damage is almost, it's certain because those positions are extremely conserved, or if they are outside of that. We have to keep in mind, also the position where a truncating variant can occur. If it occurs in the very last exon or the last 50-55 nucleotide of the second last exon, then it may escape an event called nonsense mediated decay. So if a truncation occurs over here, when it goes to be produced, the protein really does not stand alone, and it's seen by the machinery that is damaged, and therefore will be undergoing ubiquitination and degradation. So the degradation can occur even at the messenger RNA standpoint, but otherwise it's called nonsense mediated decay. But if this variant, the truncating variant, occurs here or in the last exon, it may escape nonsense mediated decay. So what does it mean? It means that you may still have a protein product. It will be truncated, it will be incomplete, but still a product. And sometimes this product is sufficient for the function, so you cannot call it pathogenic because you do not know if it is sufficient or not for the function in that particular disease. So there are genes like the lamin genes or lamin that for which loss of function is an established mechanism of disease. So if you have a truncated mutations in a gene like lamin, go for it. It's a pathogenic variant, no matter what, if it is in the canonical isoform. There are, if you look at NOM-AD, there will be some truncated variants in non-canonical lamin transcript. If you have, just to talk about tithing again, variants in tithing have been identified to, truncating variants in tithing have been identified to cause 20 to 25% of all cases of dilated cardiomyopathy. But truncated mutations, variants in tithing have been also identified in 3% of the general population. So the diff.
Can you go back
Yeah.
to previous one? So for the ones that terminated very early and then they triggered NMD.
Yeah.
And that really indicates that this particular protein is not there yet, right?
Is not there.
So meaning that it's equivalent to the gene it did not express.
It's equivalent to what we call haploinsufficiency. So you will have that one particular allele will not express the protein.
Right. But if it occurs in the later, they still may have a protein, but that protein may not be functional.
May or may not be.
May or may not be.
So yeah, so that's why you need additional evidence for that.
So is that if one of the allele triggers the degradation of this protein and then it becomes deficient, right? So is that a done deal? Tells you that that this is something will cause the disease. So it's equivalent to the gene expression is not as much, right?
Yeah, so let me give you an example again in cardiology. So lamin A, actually another gene, other two genes, myosin heavy chain 7. If we identify a truncating variance in myosin heavy chain 7, which leads very likely to nonsense mediated decay, this for hypertrophic cardiomyopathy will be classified as a variant of uncertain significance. Why is that? Because haploinsufficiency is not a mechanism for myosin heavy chain 7 in hypertrophic cardiomyopathy. Conversely, if the same type of variant is identified in another gene causing hypertrophic cardiomyopathy, myosin binding protein C3, then we will be confidently call this variant as a likely pathogenic because we know that haploinsufficiency of myosin binding C3 is a cause of the disease, while for myosin heavy chain 7, a haploinsufficiency seems to be quite tolerated. So just the interesting part of tithing is that the distribution of those truncating variants in cases versus controls are different. But when at the beginning they find that 3% of the population have a truncating variant in tithing, this was very troubling. So also, we have to keep in mind when we look at the splicing variants, not all splice variants are born equal. Of course, if we have a gene, again, like myosin heavy chain 7 that has only one known transcript, that this will lead to a alteration in splicing, even though it's still very not uncertain significance from a clinical perspective. But if you have multiple transcripts and multiple isoforms, so then a particular splice alteration may or may not be clinically significant. And so you have to identify whether this splice variant affects the most important biologically functional isoforms. The other thing is that there are a lot of genes like both dystrophin and the tithing gene where there are a lot of repetitive elements. And there could be exons that are either so-called blunt, meaning they finish in frame, or they're finishing out of frame. So depending on where the splicing variants events occurs, you can have either in-frame or out-of-frame, let's say, events. So in this case, this is the dystrophin gene, so we have multiple promoters. The rod part of the gene is highly repetitive with blunt exons. So a deletion of blunt exons can lead most likely to a milder disease called Becker muscular dystrophy, which has a more prominent cardiac disorder, less prominent skeletal disorder, versus those out-of-frame events are causing a Duchenne muscular dystrophy. So it's very important from a clinical perspective to know exactly what this, even if it's a truncating variant, even if it's a splice variant, what it does and where exactly. When we have missense variants, is also complicated. We need to know if the variant is conserved. So among the different species, we have prediction softwares, but those prediction softwares, they have an error in that are intrinsic in their algorithms. So there are multiple, software that can be used, and that's all the pros and cons of those software. But again, they don't predict effect on interacting proteins. Then there are also to be always aware that there are some type of variants that may not have what we call radical change, like the truncating variants, like in frame or non-frame shift indels. They don't change the reading frame. However, they can be pathogenic, as we saw for Delta 508. This is another variant in the ion channel. This is a sodium channel. SCN5A has 4 transmembrane domains with 6 transmembrane segments. And those segments between S5 and S6 are the pore region. S4 are the voltage sensor of the protein, of the channel. And this variant is a in-frame change in a intracellular loop. So in theory, you would say, well, maybe quite tolerated or benign. This is actually one of the first and best characterized known cause of long QT syndrome. So non-frame shift or in-frame variants can be pathogenic, but also you have to be aware of genes where you can have stretches of very similar amino acids. Like in this case, this I think is ZIG3 is a gene that is causing heterotaxy, so congenital heart defects as well. And as you can see, there are stretches of alanine or histidine, and a deletion of 1 or 2 or a duplication of 1 or 2 of those amino acids may be tolerated, as it occurs when you look at different species where there are different numbers of those amino acids. The other misconception that was put forth and finally disregarded many years ago was that silence changes could be, discarded because they were not doing much. Synonymous changes, especially, for instance, if the last nucleotide of the exon, are important place where transcriptome elements can bind. In addition to that, we have that exonic sequences like exonic splicing enhancers or silencers are also binding site for spliceosome elements. And so even a silent change can be deleterious. Oops, sorry. The other point that I wanted to point out are the branch point. There have been few branch point changes that have been identified that are very well known cause of diseases in different types of diseases, arrhythmia or breast cancer and so forth.
How do we know where is the branch branch point? If it's only a A there, there will be a lot of code.
Yeah, usually it's always at the same distance or very similar distance from the exons. We are talking about 30 something.
Yeah.
Yeah.
So every 4 nucleotide there, there will be an A, right?
See, not all A's are born equal.
That's right. That's
Even
how do we find it?
It's like everything in life. Even if it looks like the same, it's not the same. So another way that a clinical laboratory have been a powerful tool to discriminate between bystanders or tolerated changes versus likely pathogenic changes was to do a family study, to do the co-segregation analysis. So if the variant occurs in all affected members, but not in non-affected members, then we have a good chance that this is really the cause of the disease. Also, if you have 2 changes and one is in an autosomal recessive disorder, those 2 changes, you need to know the phase where those changes are coming from. If they are in cis, well, then you have a problem. If they are in trans, then you have a suggestion that those 2 variants may have a clinical bearing on the clinical phenotype. In studying the segregation analysis from the pedigrees, you need to have the largest pedigree possible. Unfortunately, right now, currently, large pedigrees are no longer as available as before, and you need many affected members and multiple generations. But you have to always keep in mind the type of disease you are trying to analyze the variant for. If it is a disease which has a very early onset, therefore studying young generations could be feasible and important. But if you have a late onset disease that is occurring in the 3rd or 4th decade of life, or like for certain type of disease like Parkinson or neurodegenerative disease, frontotemporal dementia, they may have a very much an adult onset, late onset, and therefore analyzing a young generation may not be as informative as you may have thought. So in the absence now of large families, we rely on population genetics. So if we cannot, it's like with the SNP analysis. Instead of doing linkage with few microsatellites, if you analyze millions and millions of teeny tiny data points, you may retrieve the same type of information. So getting population genetics, all those studies with case control, large GWAS studies, can be useful to determine whether a variant is overrepresented in cases versus control. So it's more present in disease than in subjects without the disease. But also keep in mind, again, there are variants with incomplete or age-dependent penetrance that can be in controls. Just to give you an example, the gene is HFE, which causes hemochromatosis. The change is histidine 63 to aspartic acid. This variant is one of the most common causes of hemochromatosis. But even in homozygosity, because it's autosomal recessive disorder, or in compound heterozygosity with the cysteine 282 to tyrosine, it can cause a disease only in a subset of the subjects that are homozygous or compound het. Usually 5 to 10%. So only 5 to 10% of subjects which are homozygous or compound heterozygous for those variants may show a clinical phenotype, which can be very severe, but so the penetrance is very low. And in the general population, those variants can be overrepresented. So we cannot always call benign something just because it's present in general population.
But do you think that is something to do with other factors that I mean, it's interaction effects, meaning that you have it, it's probably okay, but actually the other factor is, I mean, compensated function or something like that.
So we have variants that are giving susceptibility to disease. Those diseases may not show up unless a trigger happens.
Yeah.
This trigger can be the other genetic milieu, but usually it's not sufficient. Otherwise, the genetic milieu occurs from birth. So we should have it all the time. Many times it occurs as an environmental factor. So the so-called recreational drugs, which are killing people with arrhythmias, or metabolic stresses. For instance, there is a transbarcarmylase deficiency, which is causing a form of urea cycle disorder called OTC. It's a gene that is X-linked. The carrier females, they are usually non-symptomatic. But if the urea cycle can lead to metabolic stress, metabolic problem when you stress it with the over protein intake. So essentially, if a female subject that is a carrier for an OTC variant goes to have a T-bone steak and she doesn't know that she has OTC, she may have a metabolic crisis and will be showing up and she needs to go to the emergency. So we have triggers that are environmental triggers, and we include, of course, what we do. And so those needs to be sorted out. And so that's why it's sometimes we have a lot of frequency, it's skewed toward those low penetrant variants that may be just functional only with a trigger. Like QT prolonging drugs, if you have a susceptibility to QT prolongation, you take the prolonging drug, you're going to be in trouble. But not everybody will have the same trouble because those drugs are extremely common.
So do you want to define VUS?
Yeah, variant, I said before, variant of uncertain significance. Yeah. So again, common, it doesn't.
So meaning that those VUS is usually something that can cause a disease, but we don't know that yet.
Correct. We don't have sufficient evidence to determine if it is tolerated or it is pathogenic.
We just don't know yet.
We don't know yet.
Those are the suspect that is a good target for research.
Correct. Yeah. So as I said, frequency not always correspond to the ability to determine the classification of a variant. So one of the most common variants is the Delta 508. Look at this NOMAD. You have even a homozygous European, probably because he was captured before he was detected clinically, or because there are cases where Delta 508 pairs with another variant, causing a not a full-blown cystic fibrosis, but can cause can cause us a problem in sterility in males. And so that's another which may not have been detected, but it's up to 1.2% in the European population and is less frequent in other populations. So this tells you all the complexity in human disease. There are some common wisdom, elements that have been always utilized, but they have been shaken lately. So one is that, of course, in an autosomal dominant disease, AD is autosomal dominant disease, one variant is sufficient to cause a disease. When autosomal recessive, you need two changes. And the variant frequency cannot be, has always to be less frequent than the disease frequency. But again, so if the variant frequency, the expected variant frequency has to be sometimes is more than the disease frequency in cases where there are multiple genes involved. And so there are digenic or oligogenic diseases that are present in the population. But even in those so-called Mendelian disorders, monogenic Mendelian disorders, like long QT syndrome or hypertrophic cardiomyopathy, they have been identified up to 8% of subjects that were carrying more than one pathogenic variant. And in hypertrophic cardiomyopathy, up to 10%. And usually myosin binding protein C3 is a gene that often has a second, offer a second hit in those patients. But also a more complicated form of cardiomyopathy, which is the dilated cardiomyopathy, has been seen to be quite complex from a genetic standpoint. In a study of 639 patients, they have identified that 38% of those subjects were carrying two pathogenic variants, and up to 13%, 3 or more variants. So where the frequency of those variants seems to be somehow higher than the disease prevalence. So are those a bunch of low penetrants putting together and causing the disease? So is it a mutation load effect? And we don't know yet. Okay, so
do we want to take another break?
Do you need another break?
Yeah.
Yes? Okay.
I think so. If you ask them.
Right. How many more? No, 20 more slides.